Boa tarde a todos. Vamos iniciar a sessão. Primeiramente, obrigado por tê-los aqui, obrigado por participar. Meu nome é Vitor Carvalho, sou gerente da editora científica Karger. Quero agradecer a parceria com a Sociedade Brasileira de Genética Médica e Genômica e destacar que todos os associados da SBGM, vocês possuem acesso a alguns periódicos da Karger em genética. Então, só para citar alguns: Cytogenetic and Genome Research, Molecular Syndromology, Public Health Genomics, Sexual Development, Human Heredity. Todos esses títulos, vocês possuem acesso ao texto completo, basta acessar via portal da SBGM e vocês vão poder acessar esses periódicos da Karger, ok? Agora, entrando no tópico do webinar de hoje, como a inteligência artificial tem revolucionado a pesquisa de literatura genética. Obviamente, a inteligência artificial já é uma realidade em nossas vidas e cada vez mais estará presente, mas especialmente na parte de pesquisa de literatura genética, já existem algumas tecnologias realmente mudando o paradigma atual, tornando a experiência de pesquisa muito melhor e deixando essa busca de literatura muito mais assertiva, né, na encontrabilidade dos artigos científicos em genética. O nosso speaker de hoje é o Jeffrey Saffer. Ele é um pesquisador biomédico, PhD pela Yale. Há mais de 2 décadas ele está envolvido em informática biomédica e ele tem liderado esforços em inteligência artificial para pesquisa de literatura biomédica. Recebeu várias premiações, dentre elas a R&D 100 Awards, que é uma importante premiação do setor de inovação. Quero encorajá-los a compartilhar seus comentários, suas dúvidas no chat, e a gente vai endereçar ao Jeffrey Saffer na sessão de Q&A. Então, fiquem à vontade para perguntar tanto em português ou em inglês, e a gente vai repassar isso ao Jeffrey Saffer. E apenas destacar, por fim, que essa sessão será gravada e todos os participantes vão receber tanto a gravação quanto a apresentação que foi feita aqui logo após o webinar, ok? So now just switching to English. Thank you, Jeffrey. It's a pleasure to have you here. Thanks for your time and your availability. Feel free to start whenever you would like. Thank you. Hi, Jeffrey. I can't listen to you.
Okay, there we go.
Yeah, perfect.
Boa tarde. I appreciate your joining us today. As Victor has given you some background about myself, just a key point is that both myself and Vicki Burnett, the other co-founder of Quirtle, we are molecular geneticists. We understand and have suffered the pains of finding information from the literature, just like you have. And our approach to this using artificial intelligence is one that has really understood what those pains are from the perspective of experts in the field, as opposed to, say, a computer specialist building something that they think is kind of nice, but not necessarily as helpful as it needs to be. So as we get into this presentation, I think those perspectives will be fairly obvious. For the discussion today, I want to start with talking about the why artificial intelligence is needed for literature discovery. Then I'll go over some of the AI options of broad categories to help you understand what the benefits and limitations of each of those different options are. And then I'd like to take a little bit of time to introduce you to Qinsight, our own AI application that I think you'll find to be particularly useful in the area of medical genetics. So to begin, why do we have to think about AI? The big issue, of course, is that there's a huge volume of information, tens of millions of documents that are out there, actually considerably more than that, more like 50 million documents that are relevant. And it is just not possible using traditional methods to be able to get the detailed understanding of what we need for our work. The traditional searching, as I think you have probably experienced, gives you a long list of results, and it's left to you to read those, to figure out which of these documents you want. And if there's anything more than 10 or 20, that becomes a really impossible task. The second problem, of course, is that there are many irrelevant results. When you would do a search in many platforms, there's just so many irrelevant results that you say, I don't see what I want. You start changing the query in order to get to something that is meaningful on that front page. And in doing so, you throw out some perhaps critical documents, and that is definitely a problem. And even if you have the right results, which rarely happens with tools like PubMed, it is difficult to explore it. How do you know which of these things do I really want to look at? And so often we're missing a lack of context. As part of our own adaptation to the limitations of tools like PubMed and Google Scholar, we start narrowing down a query. We don't understand the big picture. And that big picture, as we as scientists certainly understand, has to include a systems level perspective. We can't just ask about, well, what is happening with this gene in this disease? Because, I don't know of any examples where a gene works all on its own. It's obviously a part of a complex system inside a cell, and having the bigger picture is a great way for us to really understand the context of what we're looking at. These failures of traditional searching is really huge. It's been estimated that the impact of missed information from the biomedical literature to the biomedical industry is over $100 billion per year. That's a huge amount, but I certainly can understand it, because in our interactions, say with some pharmaceutical companies, we have seen billion-dollar mistakes made over and over again. And these kinds of problems happen quite frequently. So it really is not just a matter of our own frustration, it's a matter of wasting money and delaying the time it takes to get improvements to healthcare to the population. A second key issue about why AI is needed has to do with the very topic that your society is interested in. It's understanding genetics. And as a prelude to talking about this, I will say this is something that AI can do. I'm not necessarily saying that AI is doing it in the way that it's been implemented in many cases. In fact, I think that is the case, that it is not well implemented. But certainly I'll be showing you a place where that is possible. So being able to understand genetics is absolutely critical. An example, the ambiguity of terms. If you search for bad, B-A-D, maybe as a molecular or medical geneticist, you're thinking about bad the gene. It also might mean not good. It might be an abbreviation or an acronym for something else, like bipolar affective disorder. Traditional tools simply cannot handle that. Those keyword-based approaches are inefficient. They do not distinguish when bad means one thing or another, and this is just one of the many reasons why you get so many irrelevant results in the list. AI, in fact, is able to do this if implemented correctly. Second problem for genetics is the huge number of variants. Bad itself has many different names, like BCL2L8 and so on. A very common gene with regard to human disease, TP53, which I'm sure you know was associated with more than half of human cancers. Our system has learned that there are over 160 different ways that gene is called in the literature. And even if you knew all those 160 plus names, to do a complicated Boolean query where you wrote them out, TP53 or TRP53 or P53 or BCC7 or blah, 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 that's just painful and not very likely that you'll do that. So to have the artificial intelligence be able to understand concepts, to have it work on what a particular concept really is and all of its relationships without you having to do that kind of complicated Boolean is really a great benefit and gives you a much better and deeper discovery. We have to face it, tools like PubMed simply do not search genetic information effectively. Going back to the TP53 example, if you do a search on PubMed for TP53, you get a different set of results than if you search for TRP53, or a different set of results if you search for the protein P53 and so on. That really is not helpful, and certainly we ought to expect more from PubMed, but that just isn't the case. It's not true for Google Scholar, and in fact, it's not true in just about every other application that I'm aware of. So these are key problems that we believe artificial intelligence can solve if applied correctly. So let's talk about some of the existing methods that are out there so that as you are looking for more effective ways to look at medical genetics literature, what you should be considering. First of all, I want to mention natural language processing, NLP. This is a simple form of artificial intelligence. It's rule-based and really is centered around the idea of a subject, a predicate, and an object. Here's a current topic. SARS-CoV-2, the virus, causes COVID-19. So there is a stated relationship here that the virus, SARS-CoV-2, is responsible for COVID-19. This set of subject, predicate, object is often referred to as a triplet, and NLP is really all about discovering these triplets. And this is a process that can be automated. An example of an application in this area, for example, is Linguamatics I2E. And the benefit of this is it's really good for basic text mining, pulling out all of these different triplets. And as you'll see in a moment, those triplets can then be applied in additional layers to give us some better understanding. The difficulties, though, of looking at these NLP triplets is that the tools that generate them, like Linguamatics I2E, are hard to use. It usually requires programming. I know from many people that have told us who have Linguamatics I2E, it can take them days to weeks to execute a query because of the programming necessary to set up that query. And that's just not effective when I have a question right now, I want to look something up, I need an answer. The other problem with NLP is that triplets are not the only representation of knowledge that occurs in literature. So a deeper form of AI may be much better at doing that. Knowledge graphs are an example of taking NLP even further. Here, the idea is to assemble all of those relationships identified by NLP and have a causal or trait connections that allow you to make some inferences. So this is really great where we're looking for indirect connections. So as an example, say this is a gene, it causes a change in this biological process, which then affects this disease, which then has this effect, and you can sort of go from gene downstream through each one of these different pieces. It sounds great, and the representations like this one can look pretty spiffy, very nice looking, but not always as helpful as we want. Examples of tools that are knowledge graphs would be causally or YEWNO, that's YEWNO. So there's a couple of things that you might want to look at. But the difficulties here of this kind of process is that it's really difficult to explore this entire map and get that global perspective. As I said, we want a systems level perspective. We don't want to just narrow in immediately to a specific gene and look downstream and see how that fits into a particular medical situation. We'd like to be able to interpret that in light of everything, but everything is in fact represented by this entire network. So you can't do that. A second problem of the knowledge graphs is there's a lack of context. We have seen examples where, say, this bubble in red is connected to this other big bubble in red, and that maybe is true, say, in an animal model. Maybe somebody said in mice we found A is connected to B. That is not necessarily the case that that's true in humans, but the knowledge graphs take all of the available information from triplets, puts it together, and you can't know, because you have no context in a map like this, what that really means. And in fact, you can get incorrect inferences. A banana is yellow, so there's a connection between banana and yellow. Yellow is a trait of jaundice, but that does not mean that bananas have anything to do with jaundice. So these are the kinds of problems that can occur. And here's one that is actually was rather interesting from the literature. Isoprenaline, is said to treat asthma. And there was a lot of literature, particularly from animal models, about that. But in fact, when that was first translated to human use, thousands of people died because of the administration of isoprel. I think that was largely a matter of dose, but again, the lack of that information not being available immediately obvious to whoever is looking at a network graph can really lead to these kinds of bad decisions. So it's a really good approach for finding these indirect connections, but you do have to worry about the lack of context and the incorrect inferences. Citation networks are another type of graph connecting many different, in this case, articles together rather than concepts. And this is great when you find a document you're interested, maybe to find others that are like it. Examples of citation networks would include Meta or SiteNet Explorer. And so this is one way you can find these related papers. So I found this, now I can use a citation network to find other papers that are related. The difficulty here is that citations are necessarily biased towards older papers. So the older papers are cited more times and end up driving connections that are not necessarily about what is most currently relevant. Those citations in a paper are not necessarily what the paper is about. For example, say in a methods section of a paper, you are using, say, a detection method that was described for a paper about something in bacteria. It has nothing to do with what is relevant to humans. And so you get some connections that are mixed that way. And all of this approach of using citations is actually an attempt to reflect the author's message in the paper, but in fact, it can't do that as well as directly understanding those messages. So again, it's got some very good use for finding related papers, but there's caution in using these kinds of networks. And again, like with the knowledge graphs, can be very difficult to explore. Deep learning is another AI method. Probably the prime example here that you may have heard about is IBM Watson. In the deep learning, The idea is to use the computer programming to automatically discover the features that are relevant for helping you make comparisons. This is really good approach for determining complex relationships, things that really are not well represented in NLP triplets and other structures that traditionally have been used, and certainly much better than keyword searching. The problem, though, is that for deep learning, you must have a training set for every case that you're interested in. As an example of deep learning, think about the classic example of having a computer look at a number of pictures. You tell it in the training set that here are some pictures of cats, and in another set of pictures you say these are not cats. The artificial intelligence deep learning is able to look through these different pictures, and based only on that training set you gave it, begin to identify the features that distinguish a cat from something that's not a cat. For a simple case like cats versus non-cats, that's pretty straightforward. You can pull together a lot of pictures and do that very quickly. It's also useful in a situation like that because cats aren't going to change very quickly. A cat today is pretty much going to look like a cat 10 years from now or even 50 years from now. On the other hand, for complex biomedical problems, there are so many different questions that you want to ask that the training can take an enormous amount of time. And our understanding is changing over that time, so that by the time you have finished the training, it's already out of date. And as you probably know, IBM Watson for Health is pretty much now defunct, and that really emphasizes the difficulty of using this kind of approach for understanding the literature. Another problem with some of these deep learning methods is the introduction of bias. There's a similar kind of problem in just in using Google, for example, or Google Scholar. The more times somebody that's done a query goes and clicks on a result, that biases the results in the future towards that paper. So that when you see results, what you see is biased according to what other people are looking at. Well, what other people look at may not have anything to do with your particular interest in having done that query. And so those kind of biases then can hide the very information that you might be looking for. So again, it can be a very powerful approach, but we do need to pay attention to those limitations and be sure that it's something we need. And in general, the deep learning simply cannot keep up with the changing face of information we need to be looking at. So sort of summarizing these limitations and touching on others, they're generally very hard to use. It took IBM 4 years to set up the deep learning system for MD Anderson Hospital at a cost of $60 million. So that's a problem. The poor support for genetics, which AI is readily able to solve, but is not implemented well in most of the AI methods that I've mentioned. Another problem that strikes us from the scientific perspective is just because somebody says this gene is involved in this disease does not mean that it's so. We all know we have to look at the papers, understand whether the experiments were done correctly, was the interpretation correct, and so on. So we should have some measure of believability or level of evidence in the information we're looking at, and that is missing in other tools that we know of, but certainly the kind of thing that we as experts recognize ought to be there. And we also might want to really focus in on some additional predictive capabilities, really take advantage of the additional insights that artificial intelligence can give us. So this is kind of what we want. So given what we want, the best thing that we want the power of AI with unprecedented discovery. We all agree with that. We need the methods that underlie this, though, to understand how we think as scientists in this field. Understand genes, understand the concepts and the disambiguation, understand the very kinds of questions that we as medical scientists want to be answering. The system needs to be as easy to use or even easier than PubMed. We'd like to be able to support asking just simple questions. Instead of doing complicated Boolean, maybe I just want to ask a simple question. We want the results to be immediate. I don't want to wait weeks to do the programming or set up a system. We want there to be a measure of relevance that makes sense. We want that level of evidence and that predictive visual analytics, as I said, to get deeper insights. So that brings me to what we have done that really distinguishes our application, Qinsight, from all the other types of AI applications that we're aware of that are presently available. We really want to move from search to discovery and true understanding and address all the different kinds of issues that I said. So with that, I'm going to switch over here and do a brief demonstration. I understand that one of the areas of interest for your society might be oncogenetics. So I'm going to do an example in that direction just to get started. And here I'm asking a question about what are the genetic factors that are associated with prostate cancer. This is a prelude to getting at more detailed information, but it's a good starting point. And it's a way to demonstrate how having a systems level perspective gives me a big picture. So to start with, you can see that we've asked a question about genetic factors associated with prostate cancer. In the results, rather than looking for terms like genetic and factors and so on, the artificial intelligence can interpret this and recognize that we're asking about, well, what are the genetic factors? So in this case, genetic factors includes genes, the protein polypeptide products from those genes, multimeric proteins like AP1, NF-kappa B that don't come from a single gene, microRNAs, long non-coding RNAs, circular RNAs, all the genetic factors we can cover at once. And in the results that you see here, it is easy for you to recognize that every one of these results has a meaningful relationship between some genetic factor, in this case androgen receptor, and prostate cancer. So we get relevant results right away. We can explore these results by seeing additional statements from the documents that show why it's relevant. We can view the abstract. It makes it very easy for us to see why these are relevant. And in this kind of relevant discovery, what the artificial intelligence is doing is to ensure that the author has said there is a meaningful relationship between some genetic factor and prostate cancer. That is much smarter than just asking whether there is an occurrence of a genetic factor in the document and occurrence of prostate cancer. And in fact, in this set of results, we have 165,000 articles that tie some genetic factor to prostate cancer. If we didn't require there to be this meaningful connection, we would have over twice that number of documents. So in fact, the noise in the system that would be present if you're doing traditional keyword searching says that half the papers, in fact, wouldn't be relevant. Of course, now this is a huge data set, and we want to be able to explore this much easier. To do that exploration, let's turn our attention to the left-hand side of this page. We can filter by date, filter by publication type. With regard to publication type, I think it's obvious from this list that we cover much more content in Qinsight than PubMed does. We have essentially all the biomedical journal articles, including a lot that are full text, including all the content from Karger publishers. We cover clinical trial information, patent information, treatment guidelines, NIH grants, so you get a glimpse of what people are working on now, even before there is a publication. News, the Toxline database, Daily Med, the drug labels, conference abstracts, and gray literature includes things like the Henry Stewart talks, authoritative presentations by university professors, but not necessarily peer-reviewed. That's why it goes there. We have filters that we can start narrowing this down in a logical way, and as you'll see in a moment, we'll be able to follow that easily. Negative statements. This is great because I think I saw up here, for example, here is a statement at the very beginning about androgen receptor is not altered in primary prostate cancer. So if we're looking for a direct connection between a gene and the cancer, this actually doesn't apply. So the idea of the negative statements is to use an artificial intelligence layer that allows us to begin to focus in on the positive statements only, or focus in on the negatives. This is really useful for doing things like, well, why does one author say this is true and another author says it's not? Does it have something to do with the patient demographics? Is it the way the protocol was carried out, and so on? And this is the only application we're aware of that allows you the opportunity to tease out those differences. Most importantly here, even though this is a huge number of results, you can understand it very easily because another layer of artificial intelligence goes through these documents and automatically discovers the concepts that are relevant to what I'm interested in. In this case, since we've asked about genetic factors, this is the list of all the genetic factors that are relevant to prostate cancer. The list is ordered according to level of evidence: androgen receptor, the kallikrein-related peptidase 3, and so on. And so this gives us a way that we can focus in on what's most likely important and work our way down. As you can see, for example, here in result number 5, a microRNA is involved in this, and this is a great way for us to explore. If we were to have done the same query on PubMed, which I did here, you can see instead of 135,000 results, there are only 6,700 results, and no way to actually explore this without reading the documents. So, clearly, and I think probably from your own frustrations of using tools like PubMed or Google Scholar, this just is not an appropriate way to get at genetic information. Coming back over here to this application, now I can immediately look and say, what's the role of P10? Click, and now the results will update, and I can start getting into a very specific example that I want. Maybe I want just the results from the last year. So we get these breadcrumbs where I can understand what filters have been applied. So I'll remove those. And let me also point out, as we go back to the results, that the application is also pointing out a couple of different forms of prostate cancer that we may want to focus in on. So having done this and thinking about the questions that you might have about, well, which of these genes might contribute, say, to susceptibility to a disease? Here I'm going to do an additional query using the also containing filter. I'll just do susceptibility. And now I'm going to get to a very focused set of results where there is a meaningful connection between a gene and prostate cancer with regard to susceptibility. So now I have a list of the genes that are most likely associated with that susceptibility. The sorting order of the results themselves is based on this believability analysis and understanding what is most likely relevant to what you're interested in. And the AI keeps up with this as you explore and keeps adapting. Now that we have done this, you can see we have an updated list of the genetic factors. These genetic factors, now we can start to explore. And here we recognize, of course, that this is over all time, and our understanding of prostate cancer and the genes associated with susceptibility is probably changing over time and will continue to change. In fact, if we go to this last year, you can see where we had androgen receptor was the top of the list. It's now dropped down, and kallikrein-related peptidase 3 has moved up. PRAC1, the small nuclear protein that wasn't at the top of the list before, is now moved up. This metastasis-associated lung adenocarcinoma gene is now moved up higher on the list, and so on. So this is a clear indication that our understanding is changing over time. So let's go back to here, remove that one, and introduce one of the visualizations that we have that helps us understand this kind of information. Recognizing how the AI can look at features and pick out information that even if I read all of these 1,800 papers, it would be very difficult for me as a human to see. We can train the artificial intelligence to look at this information, understand what the authors are actually saying, and now give us a representation of this information that does two things. In this map, let's just go up here, you can see that the genetic factors are these blue bubbles, the different forms of prostate cancer, the purple, the general concepts pink, and the actions that sort of glue these together are the gray bubbles around the outside. Focusing in here on the genetic factors, what we can see now is the relative levels that are present, where androgen receptor is the largest, then KLK3, and so on. Now we can not only see the relative levels within each group, but across the group, of course, we can see that since we've asked about susceptibility, all these papers are about that, and see these relative amounts. So that relative level is one thing that really helps us. Perhaps more importantly with this visualization, you can see, for example, here for BRCA2, there's a red border around this particular bubble. This means that based on what the authors are actually saying in these documents, the AI is able to predict that the role of BRCA2 to the susceptibility to prostate cancer is likely to increase in the future. So there's likely to be more emphasis on this versus, say, KLK3, which is a blue border, which means the interest in that is likely to decrease. Now, in this case, BRCA2 is a relatively large concept itself. And if you did the normal approach of starting with the biggest and starting to work your way down, you might have, in fact, uncovered this. In many cases, though, a concept that is emerging and is predicted to become more important may be one of these very small bubbles. And so the red border in a visualization like this is a neon sign saying, don't overlook this, even though it's low on the list. It's an opportunity to discover something that may be just emerging as a concept, and gives you something where you can really focus in on that. All of the visualizations in this application are interactive. I'm going to just now click on BRCA2. And you can see we're now down to the papers that are about BRCA2. And I can see for myself why this might be emerging as something that's becoming more important. So that while this is an artificial intelligence powered application, we like to think of this as augmented intelligence, assisting you as the expert to make the final decisions. But here now I can see exactly what's going on. Let's go back and get rid of this and take one other perspective here, showing another visualization of this application that's going to be particularly useful. Once again, even though we are experts in the field, we look at a list like this, and it is all too often we fall into the trap of thinking about what is the role of androgen receptor, what is in susceptibility, what is the role of KLK3, et cetera. And we forget that this is a complex system. So this visualization that I'm opening now, called the connections visualization, is a way that we can understand how all these different factors may interact and be relevant to the overall susceptibility to prostate cancer. In this particular visualization, the color orange off the diagonal, where we have all of the top genetic factors as both the rows and the columns, the color orange off the diagonal is a total probability that that pair of genes works in concert with regard to the susceptibility to prostate cancer. So you can see that there's a number of things here that have that connection. We can also position this by frequency and get a different perspective. But here I want to focus on this network graph, which, unlike the knowledge graphs that I mentioned before, now this network graph is specifically focused in and in the context of the question that I'm really interested in. So now I can look at this and see exactly how this fits together. And what I see is that there is a large group of genes for which overall across the entire data set, the artificial intelligence says there's either direct connections or predicted connections or implied connections, from what the author is saying, that says these may have some interrelationship with regard to the activity. And there's a number of factors that as yet have not been tied directly to another factor with regard to that susceptibility. Here again, we can explore this and say I'm interested in something like RNA SAL, and I can now see specifically what are the factors that is directly connected to. And here I'm going to click on the link between that and MSR1. And now the results give me exactly those papers that talk about the connection between these two genetic factors with regard to the susceptibilities to prostate cancer. And I can very quickly understand for myself, is this something that I want to pay attention to? But again, it's made it very clear to me that this is not just acting on its own, but there's some interactions that are in fact very important for this susceptibility. So this has been a quick introduction into how all that works with regard to looking at oncogenetics and susceptibility. I might point out that instead of susceptibility, let's remove those filters. Another way to look at this from a medical genetics perspective might be to ask about ethnicity. So here again, it gives us something about the genetic backgrounds of individuals, in this case with regard to larger groups. So we can see, for example, that SUN2 may be prominent in Japanese populations, GRPR in Koreans, etc. So there are a lot of different ways to explore these kinds of questions in Qinsight, and it really just follows the natural exploratory process that we wish we had in the tools like PubMed, but with a whole lot of power and a lot more specificity. And then just to sort of turn this around and give another example application direction, where you might be interested in genetics of rare diseases. Here I might be interested in a particular disease, and I'm just going to ask a simple question about what rare diseases have been connected to the glucosyl ceramidase beta. So we'll do this search. And again, here we have a list of results where there is a meaningful connection between GBA and some rare disease. Every single one of these is highly relevant. If we didn't require that meaningful connection, even if PubMed could find all the different concepts around rare disease and all the different names for GBA, it would have been like only 1 out of 3 documents would have been relevant. So the AI saves you so much time, gets you right to where you want, and now I can immediately see what diseases GBA might be relevant to: Parkinson's, Gaucher's, Lewy body disease, and so on. So this is all just a great way to really experience the depth of the literature in a way that is much more supportive of the kind of genetic information that your society is interested in. So with that, let me point out that we give you the opportunity to try Qinsight for yourself. You will get more information about this in a follow-up email, but you can go to this link. There's a special code for your society. When you sign up, you will have 1 month access to Qinsight for free, and definitely try it out and give it, get the experience of better discovery. So let me now stop here and give you a chance to ask any questions you might have about AI in general or Qinsight specifically.
Thanks, Jeffrey. So please feel free just to write down your questions in the chat or the question section, and we'll be able to address here, either in Portuguese or English, whatever you prefer. Just so that the audience start warming up, perhaps, I would like to ask, does the journal impact factor influences somehow on the results of a search in Qinsight, for example, or how is that?
Great question, Victor. The answer is there is, for the definition of results, the impact factor is not included. Some of the most unbelievable papers have been published in the most prestigious journals, and there have been very many important papers that have been published in low impact journals. So the artificial intelligence starts by looking at this from a pure perspective, understanding believability, and putting the information there that is most relevant for you. Let me just go back to the application though for a moment and show you that there is an option. If you want to limit the results to high impact journals, you can do so. So you can slide this along, and this will apply just to the journal articles. You can say, go up here, and I only want the top 10% of journals by impact factor. Now I've got a different set of results. So that tool is there if you want to focus that way, but a priori, very important papers can appear in lower impact journals, and bad papers can appear in high impact journals.
Thank you.
Well, in the absence of questions, let me just take a moment to point out a couple of other things, because I do hope you will use the application. You can get direct access to articles that are of interest. You'll see there's a PDF link, or there may be an open access icon that you can click on and get directly to the PDF. You can export the results and save it in tools like EndNote, Mendeley, Zotero, all the kinds of interactive and connections that you might expect are available here. And while I did talk about the results being sorted by relevance. There is an option to sort by date to put the most recent results at the top. And in this case, you can see a very recent paper here. And in fact, in many cases, you also see a electronic version ahead of print being mentioned. But that has all the kinds of interactions that you would expect, along with all of the power of AI. And there are other visualizations here that I did not demonstrate today that I hope you will explore when you get into the application. So if there is nothing more, let me say, Brigadu. I appreciate your taking time to join us today. And this lovely picture from Praia de Sancho, where I was just a few days ago. I took this picture and had a lovely time in Brazil.
I think we had a questions just. So Mariana asked if it's possible to do a search in Qinsight beyond genetic literature. And
absolutely, there is no limit to what this application can do. I obviously did a genetic example given the society's focus. This application is used in every aspect of life science, not just even biomedical. In the biomedical field, of course, target discovery, thinking about pharmaceuticals, understanding impacts of compounds, health economics and outcomes research, competitive intelligence, new uses for existing drugs, everything that you would want to do in any application in this field, you are able to do here. And just to give you a sense, there's a set of capabilities in this application that we call power terms that may be the quickest way where I can sort of show you some of the depth of what this does. It has an understanding of anatomy and biological processes, biomarkers and compounds, different categories, diseases, signs and symptoms, drugs, genetic factors, which I've obviously done, including very specific subclasses like GPCRs you can ask about, methods, organisms, organizations, what companies are working in what area, actions, it understands verbs as well as nouns. Things like risk factors, I mentioned health economics, disease registries, geographic regions. It's very broad applicability, and we're not aware of any field that this isn't going to be useful for.
Any other questions, comments, observations? Please feel free just to write down. So everyone will receive the presentation by email, and then you have the code over there to register yourself for a personal trial, get to know better this platform and the potentialities that it has regarding genetics or any other biomedical literature research. And also, well, if during the trial, if you have any questions or comments, please feel free just to send me, even if it's a technical question. I'll be happy to ask Jeffrey or any other technical person help to answer. so that you can get the most out of the platform during your researches. I guess we don't have any other questions.
Okay, ciao.
Thank you once more, Jeffrey. Thanks, everyone. And, well, we'll keep in touch. Tchau, tchau. Boa tarde.